Hi, everyone. Welcome to our course, Systems Biology of Aging. Aging here and there. Aging everywhere. Today is lecture 7, whose topic's really great. It's about longevity as it's reflected in your plate. There's feedback loops and circuits that are beautiful to watch. And today we'll study them a lot, but not too much. Aging here and there. Aging everywhere. Aging everywhere. Aging. Thank you. Okay, so what a delicious topic, longevity and nutrition. Let's take a nice deep sigh of relief. So last time we talked about exercise and longevity. And by the way, did anybody do the exercise hour? Yeah. How was it? How was it? What did you do? No. The tennis. How was it? And you? What? HIIT. HIIT. How was it? Okay. Keep doing it. So exercise is well supported by research to extend lifespan, prevent diseases all around, all diseases almost, age-related diseases, improve mood. It's really kind of a miracle drug, you can say. The only one we have. Nutrition is not like that because it's much harder to study nutrition in human beings. And there are some things that are clear, but I want to say off the bat that there's a lot of polarized opinions about diets and things like, does coffee cause cancer? Is what's better, paleo diet, keto diet, low carb diets, Mediterranean diet? And there's lots like warring camps, very polarized, ideological. The more polarized, the less good the evidence. And why is that? It's because nutrition science it has two main arms. One is epidemiology, which is basically asking people what they eat and seeing their health after factoring out everything you can think of, education, etc. And experiments. So experiments are hard to do with human beings because human beings, you have to keep people. First of all, when people report what they ate, it's very unreliable for some reason. Even the apps, they help. And you have to keep people in a metabolic ward in hospitals, see what they left over from their meal. You can do it for a small number of people for a short amount of time, but there's still some data from experiments that I'll tell you about. Epidemiological studies that you read about with poor reporting in the newspaper, like eating 20 almonds a day reduces your risk of cancer by 17%, are very flawed. And the reason is because of bias. So they tend to have small effect size, less than 1.5 hazard ratio, and opposite direction. Unlike exercises, which is always good for you, the question is a twofold or threefold reduction in health. Here it's conflicted directions. Because there is biases like what's called the healthy user bias. So if you're healthy, you do things differently than if you're not healthy. So like low socioeconomic status, you might be exposed to junk food and also be unhealthy for other reasons. So these confounding factors, or healthy, rich people, exercise, and they can shop in the outside of the supermarket where the healthy food is. You don't have to go into the inside where the processed food is, etc. The best study, I'll just give you an example, about Mediterranean diet. It's called Predimed. It comes from Spain. 7,000 people, randomized into 3 arms. One arm got 1 liter of extra virgin olive oil every week, thinking this will make higher chances that they'll cook with oil, eat oil-related food, olive oil. The other group got some really nice nuts. And the third group, the control group, nuts every week, and it was told to eat a low-carb diet and to avoid fish and other things like that, which are in the Mediterranean diet. And this study was planned for, 5 years. It had to be stopped in the middle And the people there were high risk. They're like type 2 diabetes, high risk of heart disease, etc. They had to stop after a few years because people in the control group had 30% more severe cardiovascular events, heart attacks. So it was deemed unethical to continue with the control group. The one that is supposed to eat low carb. So the olive oil in the nut group had 30% less cardiovascular. So that means Mediterranean diet is good, right?
It's complicated.
Why? Why is it complicated?
Like there are a lot of variables like that you don't know how to consider.
That's why you do the randomized, right? But the one criticism of the study, just to give you an example of the complexity, of course, you said doing epidemiological studies is very, very important and very complicated. It's just complex. It's not that the scientists are bad or something. It's just very complex. It turns out that there's something called performance bias. In the olive oil and nut arms, they met the experimenters much more than in the control arm. And meeting experimenters makes you more mindful, let's say, of what you eat. That's a very well-known effect. So, but that's like considered one of the best epidemiological studies. Right? So in this lecture, I'll not talk about what's not well understood. I'll try to tell you about what's understood. Okay? And what's understood is one, don't eat too many or too few calories. So don't be undernourished or overnourished. And I'll just give you an example. How do we measure this? So it's all energy balance. Like you eat calories more than your basal metabolic rate, which is the energy your body spends just to exist. Just like your parents are supposed to love you just because you exist. Basal metabolic rate is just because you exist. Plus the exercise you do. Like if you run 10 kilometers, 700 calories. Usually the exercise and activity we do are less than our basal metabolic rate. That's the majority. So there's an energy balance. We'll talk about that. Okay? Energy balance. And the fat stored in your body is if you have excess, more calories than your energy expenditure. So this is measured in a lot of times for research in what's called body mass index, or BMI. How many people heard about BMI? Very famous, right? How do you calculate it? It's your weight divided by your height in meters squared. Why height in meters squared? Turn to each other and discuss. Why height in meters squared? Here and share. Talk. Why is this even a question? אתה יכול לפנות מאחוריך. You have someone behind you. Turn to someone behind you over there. Okay. Thank you. Okay. Anyone have a guess why meter squared? Why is this even a question? What's the natural thing to put there? Kilogram per meter? Huh? Meter cube, because our volume, density. Why meter square? Anybody have a guess? It's okay to say even things that are silly.
Maybe because you do not scale, like I don't know, your width does not scale with height.
So the width does not scale with height. So when you look at, that's correct. When you look at human data, the weight goes as meter squared, first of all. So when you compare this, it's like your weight compared to the weight the average person with your height has. And you can also think about it as width. So if you height and breadth scale, but your width doesn't, so it's like density times width. Now, BMI doesn't take into account density. So if we have fat, a lot of muscle, we might have high BMI. But actually, so that's a side effect. So you also need to know your lean mass to fat mass ratio, which can be measured by imaging approaches and other approaches. But BMI, so it's not good for an individual, it's good for a population. So you can talk about a population with high BMI, low BMI. But if your own BMI, we also need to know your muscle to fat ratio to understand your health. Okay, so it's very important. But on populations, you can do something cool like plot BMI versus risk of something, let's say death. Okay? And what you get is this graph. That's why I'm saying don't eat too little or too few calories. So what's going on here? This is about 22. Between 20 and 25, this is considered normal. 25 to 30 is considered overweight. By the way, I'm 90 divided by 1.85 squared equals 26. I'm here. Okay. And 30 plus is considered obese. Okay. I hope you find this interesting. And I'm sure you're calculating your own BMI now, right? So each 3 kilos is about 1 BMI point when you're 1.85 m. Yeah? How much did you get? What's your BMI?
25.
25. Great. Okay. Now what happens with age is interesting also. Let's call this the optimal BMI. Optimal BMI because it's the lowest risk. So with age, it moves like this. Moves the optimal BMI moves with age. So this, let's say maybe 50 to 60. This will be, let's be 70 to 80. That's what happens. Maybe I drew it wrong. It's like this more. Well, anyway, it's in the lecture notes. And so, like, it's good when you're old to be a little more full, because the down arm of it, like the low BMI arm, is associated with frailty. So a lot of diseases, for example, you don't want to lose weight, but you lose weight. So you can say that you don't have the reserves. This part, the low part, is quite steep. And there's even a study on Israeli 18-year-olds that showed if you're low BMI when you're 18, you have higher risk of heart attack at 50 and things like that. So it's not good to be low, not good to be fat. So not too many calories, not too few calories. And also, if I write here diseases, it's very interesting that obesity and also low associates with lots of diseases. There are several kinds of cancer, for example, liver cancer, pancreatic cancer, like deadly cancers, that are affected by obesity. Of course, diabetes. And I'm talking about over here, 8-fold hazard ratio for liver cancer. Only happens after you have fatty liver disease, cirrhosis, fibrosis.
What's the disparity between male and female?
There is. It's small, but females are a little bit higher. It's similar but higher, but that's in the lecture notes.
I'm confused about the age, the difference between the two graphs based on age. Didn't you say it's more dangerous to be underweight when you're older? That seems to be the opposite of what the graph is.
No, and what I'm saying is that the optimal weight is 27. 27, let's say, somewhere here. So you say, oh, this person is overweight.
So I'm talking about the left arm of the graph.
Oh, the left arm of the graph.
Yes. Well, I would expect it to be steeper when you're older.
Oh, yeah. That's the way it looks. I don't know how to explain it. I think there's lots of other risks of death, not only BMI when you're older. Yeah.
You say that higher BMI is a cause of disease, but weight in itself is not a cause, it's a result of a way of life. So maybe it's not the weight, it's whatever
Okay.
led to it.
So yeah, you're right. This is just an association. So let's talk a little bit about the biology of how obesity mechanistically causes diseases. Right? It's also very interesting. So when we have excess calories, we evolved to be in a situation where that we should immediately store it as fat because that wasn't so common in the evolutionary past, presumably. So you store it in fat. Now, fat cells, called adipocytes, are in a nice layer underneath the dermis of your skin. There's a nice layer on here, glutes, legs, right? That's called subcutaneous fat. It's great. It's not bad to store fat there. It's very healthy to store fat there. The thing is, you don't get more fat cells. Each fat cell grows, and it can grow to 1,000 times bigger than a normal cell. It looks like a bottle of oil. And that can't grow no more. And then there's an overflow. And fat gets stored in visceral fat, in inner organs, like the liver, the muscle, in the form of lipid droplets. When that happens, the body responds as if it's a damage causing inflammation. It also causes insulin resistance. The signals telling the body to take up sugar and store it as fat, the body says, something's wrong, stop with this. This hormone becomes less effective, and that is pushing you to diabetes. This inflammation in the liver, fatty liver disease, pushing you to liver cancer. The fatty pancreas, to pancreatic cancer, and et cetera, et cetera. So the fat also causes disbalanced fats in your blood that cause but maybe it's also time to reveal. So we have the subcutaneous, which is good, and then we have the visceral. This is the liver, right? Visceral fat, not good. Inflammation, fibrosis, insulin resistance. And that's why obesity is a cause of a lot of diseases. And Tomer?
Yeah, question. So, if you have the same number of fat cells, what happens if you do surgery to remove fat? In that case, they don't exist anymore, and you initially store it in the organs.
So the question is, if you do fat suction, then you reduce your storage capacity, and you overflow to visceral fat. Also, as we'll see, you increase your hunger. Unfortunately, we'll see that from the feedback loop, where more fat reduces your food-seeking behavior. So that's a side of.
I have a question about the model of how do you get all the age-related diseases. Now we are talking about other components to why you should have why would you get liver cancer and stuff like that because of that. And we had a model that said that when would you get liver
Cancer?
Sorry, if you cross some threshold.
Right.
So what is the result? Is the threshold go down or?
Yeah. So we had this equation where cancer cells proliferate and are removed by the immune system, and we get cancer when here we put in senescent cells, and they increase proliferation, reduce removal. That's when you get cancer, right? So this processes here, inflammation, fibrosis, they generally increase the advantage of cancer cells.
Mm-hmm.
And also when you have fibrosis, fibrosis means scarring. The liver, what happens in 30% of people is that they have fatty liver. Some fraction of them, it becomes inflamed liver. Then the liver becomes scarred. That used to be only seen in alcoholics. Now it's seen in so many people because of nutrition. Scar tissue is very stiff, and it creates mechanical environment for cells to undergo what's called metaplasia. It's to transform themselves, open up their chromatin, and become very prone to cancer. So you don't see liver cancer without scarred liver. It used to be also hepatitis, which gives you scarred liver. So it's basically let's see. Let's analyze it because this is proliferation. This is removal. So I'm saying it's increasing proliferation. So it's effectively reducing the X critical. In fact, most people are just not susceptible to liver cancer. And these people are. Only the people with scarred liver are. So that's one example, I think, of how susceptibility might work. Or if you're alcoholic and your liver is scarred. In each disease, we need to figure that out. That's a task for systems aging in the future. Your generation. The voice of your generation. Okay. So that's why fat is deadly. Where was I? Excess calories. So don't read it. Okay. Now you can say, okay, we're going to talk a lot about how not to eat too many calories and what sets our weight. Why am I 90 kilos? You're different. Why are you BMI 25 for a long time maybe? Your weight is kind of constant. I don't know, in your age. Sometimes, where is it written? Where is it written? We'll talk about that. What to eat, right? The composition matters less. So this is where the diets are, right? It's important to have more than 1 gram per kilogram protein. So here we talk about macronutrient, 4 macronutrients: protein, carbohydrates, fat, and alcohol. Alcohol, it looks like there's no dose that's healthy, even though that was debated a lot. That looks actually from Mendelian randomization. Carbs and fat, less important unless you have metabolic disorder. You need low carb. And protein is very important. So if you don't eat enough protein, you don't have enough muscle mass. We talked about it last time. You need exercise and muscle mass so that when you grow older, you lose muscle mass, muscle power, you're frail, and you can't do the things you want to do. You need your micronutrients. So you need to make sure you're getting your vitamins and minerals, of course. You know about that. And that's it. Don't eat toxins. I see I have very many roles in this course. Like yesterday, last time I was an exercise coach. Now I'm a nutritionist. It's a lot of fun. Of course, I'm not a nutritionist. It's just telling you what the research, the best I know is. And I really like science communicators like Peter Attia's book, Outlive, and he has some chapters on nutrition. All right, now let's get to the fascinating world of how not to eat too many calories. So, and again, I want to build towards a conceptual model of the weight set point. I just want to tell you that there are 3 strategies. All diets can be grouped into 3 strategies. The 3 strategies are caloric restriction, also known as calorie counting, dietary restriction, like don't eat pizza, don't eat broccoli, time restriction, like only eat 6 hours a day and fast 18 hours a day. Those are the 3 strategies. Caloric restriction, counting calories, is the most effective. Unfortunately, it's the most efficacious, which means if you can do it, it will work, like athletes. It's not effective in the sense that most people can't do it. Cannot calorie count to save their life. I mean, save their life. Type 2 diabetes, once you have it, glucose above 140. if you start exercising and reducing your calories, it's reversible and curable. But very hard for people to do that. So this is really hard. And I also want to say, in the context of longevity, caloric restriction is very famous because caloric restriction you can do to animals, right? In animals, in almost all the experiments, not all of them, extends the lifespan. So what do I mean caloric restriction? It's less than 75% of ad libitum is a word which means you can eat as much as you want. So you give the mouse some mouse feed and you see how much they eat. Now you force them to eat 25% less, or 30% less, or 40% less. But you make sure that they have all their macronutrients and all their micronutrients. So they're not malnourished. They have less calories. Explain myself? And across the tree of life, I need help. I need help. Maybe one of you guys can bring me a nice, strong black marker.
In your office?
Yeah. You have one?
I'm not a black one.
What color do you have?
Blue and
Korea? Yeah, so give me what you have. You're going to go to my office to get it?
I have red.
Huh?
Red and blue.
Blue is better. Red is... Okay, we did it. Let's see if it works. So this is age, and this is survival. Starts from 100%, and we all know this graph by now, right? So you take some animals, control animals. This is their 250. And now you do caloric restriction. And their T50 grows. This is median lifespan increases. By how much? It depends on the species. The shorter lived the species, the more you can extend. So yeast growing on less than 0.5% glucose. Worms, let's say worms, you can probably extend that in a factor of 2, their lifespan. Flies, a little bit less. Mice by 10% or something like that. And then monkeys, there's 2 studies. In one, it worked, and the other one, it didn't. And the difference is fascinating. I'll tell you about that. We'll talk about dietary restriction. Because we're a mathematical course, we like to look at the shape. I told you about exercise, if you remember last time. This is the normal people. And this is the Olympians. Remember? Perhaps you can tell a difference between this graph and this graph. In both of them, the T50 increased, but there's a difference. So the maximum lifespan, let's say the 95 percentile, also increased. But here not. And this again is seen in many, many organisms. So what? So in the SR model that we're working with, it means a lot because it can tell us which parameter caloric restriction is working on. Is it production, houses, removal, trucks, threshold, noise? So as you remember, in this case, we thought it was the threshold, X critical. In this case, the only parameter in the SR model that could give you this shape, because this this data shows scaling. So if I put here age divided by T50, so now you reduce to 50% at 1, the two curves collapse on top of each other, approximately. And the only way to get that is by stretching time, like a rubber band. Houses. So, as if caloric restriction makes the houses either built slower or less toxic. Now, this makes sense philosophically, known since the 1920s, that people were thinking maybe caloric restriction reduces the rate of living. The candle burns more slowly. Metabolic processes are slower, so maybe it's like economy is slower, so you buy less stuff in the houses and you throw less out. It was figured out in worms, flies, etc., that they have a common biological pathway that causes this. It has to do actually with two things: reduction of making new proteins, that is to say, the inhibition of the ribosomes, and upregulation of recycling programs called autophagy, where the cell surrounds proteins with the membrane and eats them up, recycles them to amino acids. So it's reuse, reduce, recycle. So the houses are making less garbage. And the mutations for biologists out here, this was one of the first aging mutations in worms discovered by Cynthia Kenyon. This was amazing because it shows how theory can be really wrong. Evolutionary theory of aging in the 1950s, 60s, 70s, 80s said aging cannot be changed by a single gene mutation. It's a multi-gene thing for different theoretical reasons. But Cynthia Kenyon, David Gems, and these pioneers found mutations in worms that makes them live 3 times longer. Single amino acid, you change a single letter in the DNA, and they live 3 times longer. And that was groundbreaking in the 90s. Because suddenly it opened up a field of like genetics of aging. We'll have a whole lecture about genetics of aging. A single DNA letter makes the worm live 2 times longer. And the mutations are, and this is one of the famous ones, are in this pathway in the cell which senses nutrients based on hormones like IGF-1, which is insulin-like growth factor that's conserved worms, flies, and talks with a machine called mTOR that when you're starved, it stops making new proteins by inhibiting the ribosome, by phosphorylating ribosomal RNA, and activates this autophagy recycle. And the evolutionary reasoning is that the worm, if it's starved, it says, okay, I'm not going to lay my eggs right now. I'm going to turn on all my defenses so I can live a few more days to reach a situation where there's food and my progeny will have a chance. And it seems that all organisms have that logic, and even use the same molecules, the mTOR, the IGF-1, to make that decision. And so it's not longevity, it's stress resistance, you can say, now to survive. But then worms stop moving, and most of their life they spend actually as disabled worms. But that extra resistance that they have carries them and translates into longevity. It's not selection for longevity, it's selection for something else. That's the take home from this. There's a beautiful book about that called Why We Age, How We Age, by Kathleen Murphy, who was one of the students of Cynthia Kenyon, and she proved a lot of interesting things. Like she was working on neurodegeneration in worms and learning in worms with aging. This book is a popular science book that if you want to learn about model organisms, how important they are, because a lot of times these genes are found then in mice, and then the mice people say we discovered, but a lot of what we know in aging comes from yeast and worms and flies. Very important. But it's somehow conserved to humans. So human beings, so I'll tell you in the genetics of aging, but centenarians, people live to 100, you look at their genomes and you ask what's in common, and one of the common things is mutations in IGF-1 receptor, even in humans. So, and many, many other lines of evidence. So it's really interesting how the same molecules control presumably the same evolution decisions, because there's tasks that all organisms need. Like, for example, it's tough now, I need to protect myself to reproduce later. That's like a fact of life. And that's like the wonder of biology, right? Didn't have to be the same molecules, didn't have to. If you disrupt DEF2, if you mutate DEF2 and they live longer, and you do caloric restriction, it no longer extends further, proving that this is the pathway. See what I mean? That's the way you can say. Whereas other ways of living longer are additive with caloric restriction. They're not the pathway. So, yeah.
Are those worms have problems with reproduction in general, like, or do they manage like?
Oh, yeah. So the question is, do these worms that live longer, are they fakakte? So in this book, that's her life's work, is to show that actually they're amazing. They look healthy. They move longer. They reproduce longer. They won't compete. They won't outcompete wild-type worms because in nature they're not flexible. So if the situation is good, the ones that are fast reproducing are going to win them out. So they're not flexible. They're locked on one strategy, but they look good, really good. And I told you maybe that there's an experiment recently where you let the worms live to very old age, where already 75% have died, and now you turn on the mutation using genetic tricks, and then you still get the lifespan extension. By that time, they look bad, but the stress resistance carries them forward to double their lifespan. So that's also very interesting.
You said that after a given mutation, if you do the calorie restriction, it's no longer exceed the life.
It doesn't improve more, yeah.
So it inhibits the same pathway. So do you mean that when you do calorie restriction, it's also like mutating the sensor?
It's like mutating because this mutation activates this pathway to the state of saying I'm starved. So if you also starve, there's another interesting mutation in worms called E2, where their mouth is defective and they live longer. And it turns out that's because they can't eat. But if you give them smaller bacteria than E. coli to eat, they don't live longer. So it's really like, again, also in genetics, there are confounding factors, etc.
But we said that mice that eat less calories looks better. So
Mice that eat less calories are thinner and have impaired immunity. Worms that eat less, I mean, these worms, DEF2, they don't eat less, they just think they're starved. But worms that eat less, I don't know what's going on. We can, a lot of people spend their life looking at worms' age, which is very important, by the way. Okay, so that's about caloric restriction. Check. Dietary restriction. So again, I said there's a lot. This is where a lot of the polarized tweeter battles are. Dietary restriction. You can. I believe the experiments done in humans. So there's this really nice work by Kevin Hall recently in NIH. He locks people up in the hospital metabolic ward and gives them really well-controlled, very similar diets that have something changed. So for example, one experiment, ultra-processed foods. So we eat a lot of processed food like cheese, but ultra-processed food is like, industrial food packed with lipid, like high-calorie, engineered fats, high fructose, all the stuff, all the delicious stuff. And you put people for a few weeks, and you have a control very carefully that is as similar as possible in terms of calories, the macronutrients, but made out of broccoli. and you get, this is the ultra-processed food, this is the whole food. You very quickly put on a few kilos, meaning that ultra-processed food is more delicious and you have less, you feel more hungry when you eat it. So we'll try to understand what's going on here with the weight set point. And a similar graph showed that low carb versus low fat, low fat, low carb, that having a lot of sugar in your diet is also delicious, and you also eat more. So those kind of things are important to know for that. So I would say it's probably founded to say that you should reduce ultra-processed food, and if you want to lose weight, think about carbs in terms of appetite. You guys are so fascinated. I love it. This, again, is a lecture that's amazing to prepare. I wish you guys to be teachers and teach stuff like this. Yeah. Tomer. By the way, Tomer, is a master's student or soon to be PhD student in my group. And I'm going to teach your stuff in the upcoming lecture. Yeah.
Can it be that the ultra-processed group is just trying to compensate for missing micronutrients?
Yeah, these guys are like expert nutritionists. You're talking about micronutrients. They made sure the micronutrients, macronutrients, everything is as similar as possible because it's an experiment. And you can see what they left over, see what they ate, and you don't have to trust their self-reports. Nave is also a master's student in my group.
You said they ate.
Tomer comes from biology, Nave comes from physics.
You said that they ate more. Is it like, because the graph is how much fat they gained. So we know, like, they tested that they actually ate more. That's the reason.
Yeah.
Like the stuff. Yeah.
Yes. And that's going to become very relevant when we talk about the feedback loop. So where are we doing? We did the experiment, calories, composition, Predimed, BMI, calorie restriction, scaling, dietary restriction, monkeys. Okay. There's 2 caloric restriction studies in monkeys started in the 80s, and it took them a long time because monkeys live 20, 30 years, rhesus monkeys. They got different results. The headline was, reducing calories extends lifespan in a primate. The second headline was, reducing calories does not extend lifespan. Then the 2 groups, run by 2 female scientists, by the way, got together and made a third paper, very thoroughly examining the differences between the 2 experiments and harmonizing it. And there's a difference that's very important. It wasn't intentional, and it teaches us a lot. The difference was what kind of food they gave the monkeys. So the Wisconsin study, where there was lifespan expansion, gave them commercial monkey feed, ultra-processed, 28% sugar. The NIH study had in-house nutritionists prepare monkey food with the same macronutrients, also had a lot of carbohydrates, but they came from whole grains that they want to say they were sitting and like singing, chanting, boom, boom, whatever. So the interpretation is that the monkeys eating junk food, by the way, ate 10% more calories because they could eat as much as they want. If you restrict them, they're healthier and they live longer. But the monkeys already eating at the salad bar, it doesn't matter if you restrict. They stay as healthy. So if you're already eating at the salad bar, calorie restriction probably not going to help you if you extrapolate from this experiment. But if you're eating junk food, calorie restriction going to help you. And indeed, the junk food monkeys had 7 times more metabolic disorder than the whole food control monkeys, etc.
What do you compare them to? It sounds like you compare them to how long they would live if they want to eat this.
Yeah.
You say,
So you have junk food as much as you want versus 25% restricted junk food, and salad bar as much as you want and 25% restricted.
So the monkey that ate 25% less, 25% less junk food lived longer
And the
than
monkey.
monkeys who eat junk food.
Yeah.
They compared.
Yeah, that's right. Each study compared to their own control.
So it doesn't sound like a mistake. It sounds like they know they're feeding him junk food.
Yeah, they said, of course they're feeding him junk food. I'm calling it junk food. It's ultra-processed commercial monkey feed. They said that this is more similar to what's called the SAD, the standard American diet. So that's why they did it. But this, I'm saying what was not intentional is to do two studies with this small difference. That's what was not intentional. But that's how science works a lot of times, right?
So how do we debate it? Like, why do it work only on high fat, high sugar diet?
Because if you eat a high sugar diet, it makes you unhealthy and you die sooner. And restricting those calories, prevents that. But if you're eating at the salad bar, as much as you want, you're healthy. At least if you're a monkey at NIH. So again, extrapolating to human, you have to do epidemiology.
What happens in mice if they eat in the salad bar? So the calorie restriction still helps.
I don't know. I don't know the answer. Okay, 3:02. Let's take a break. I want to tell you about some interesting questions I heard in the break. So one thing is I realized that I really confused you with using caloric restriction for two different things. So these are three strategies for dieting. So if you're eating too many calories and you want to reduce them, you can do caloric restriction, right? So clearly, if you're here, you want to do caloric restriction and go to here. And maybe that's like the Wisconsin monkeys. But there's another meaning. Caloric restriction in the field of longevity means restricting your diet so that you eat too little, I would say. Like the salad bar monkeys, right? Which were maybe here, move to here, or here, move to here, or something like that. Very small chance difference. They couldn't detect it. But if you're here, and you're going to restrict your calories to here, I would say it's foolish. So that's the caloric restriction from the point of view of longevity. So for dieting, it's better to actually call this calorie counting, because that's what it is. It's calorie counting, Weight Watchers, form of diet. And so that was one interesting comment. Another interesting comment was about the ethnic dependence of this. So in Asian, South Asian countries, the curve is shifted to lower BMI. There's two phenomena there. One is that less carrying capacity of the subcutaneous fat means it's quicker. You can spill over into visceral fat. So there's an epidemic in South Asia of people who are very lean, but they get type 2 diabetes, very aggressive type 2 diabetes. And that one aspect is the spillover, and another one has to do with epigenetics. When you're young, like baby, and in utero, you sample the nutrition state, and that causes epigenetic changes in your DNA that don't go away, and set your body to either what's called a thrifty set point, which means I'm going to store a lot of. Once I get a calorie, I'm going to store it because the world has very few calories, or the opposite. You say it. And apparently, a generation that went from malnutrition to Western diet very quickly has this. Their set point is in the wrong place for their diet. So that happens in South Asia, also in Pacific Islands. Pacific Islands are historically locales where you have a lot of unpredictable food supply. You can have famines and droughts and stuff like that. So the population there evolved apparently to have a thrifty genome, And now when Western diet came, you can have an island population with 90% obesity and diabetes. So that's a huge problem for certain ethnic groups. So maybe that's just like starting to touch on the, we talked about male, female, ethnic. When we do experiments only on white males, like it was done until 20 years ago, and also only on male mice, because female mice were more variable, difficult, and then you can get the wrong answer. So we have to do that medicine to sample the population equitably. All right, now we're going to change gear and ask about the weight set point. Erase the broccoli. Does it look like broccoli? Okay, great. All right, so here's the song. Have you ever wondered why your weight stays nearly constant? Give or take 5 kilos over decades, it's the same. Of course, there are exceptions where we really fluctuate, but overall, it seems there's a set point for your weight. So if you want to know the answer, and you have a curious mind, let me take you by the hand and walk you through the leptin circuit. I'll show you something that'll help you understand. Okay, so how does our weight stay nearly constant? So I'm going to talk particularly about fat. Is regulated by a feedback loop. So I'm going to go into feedback loops. This is a recurring motif in physiology and in biology. Basically, if you have more fat, you tend to seek food less and have less appetite. I don't mean, some people say, but people who are fat eat more. What are you talking about? I think for a given person, if I magically change your fat, you're going to eat less the next day. How does that work? Fat in our body secretes a hormone called leptin that's received by cells in the brain, specifically the hypothalamus, and that reduces our appetite and our food-seeking behavior. So more fat, more hormone secreted into the blood. This hormone has half-life of about an hour. It's made and reduced. Percent fat, leptin. There's a relationship, there's some variation. It's like quadratic, basically. But this hormone reports for fat. How was it discovered? Obese mice, 2.5 times bigger. So like a person weighing 250 kilos, eating 6 times more, like a person eating 18 meals a day. And what's different about them? Hormone leptin does not exist in these mice. It was mutated, or the leptin receptor. People thought leptin would be a wonder drug to inject them into obese people. It didn't work, except for about 10 people a year on Earth that are born without the leptin gene, and they obese, they can't stop eating. And leptin can save their life because it usually dies children because of infections, because this also affects the immune system. But obese people are leptin resistant. We'll talk about that. So it's not a drug, but very important to control our weight set point. All right. So what's going on here? We have 2 arms here, right? Fat. Let's call it food intake. U and fat F. Food intake, of course, increases fat. More we eat, more energy, more stores in fat. And fat, through this hormone leptin, inhibits food intake. That's a negative feedback loop. I want to analyze it with you with a very nice graphical tool that's good for any feedback loop in physiology. And that is by we're going to take each arm separately, analyze it, and then put them together and let them do their thing, okay? So this is the important graph here. This is food intake in a day. Let's say grams right now. We'll talk about the composition later. Yeah?
So when you say fat in this context, are you referring to the subcutaneous fat or the visceral fat?
It's the sum of the subcutaneous and the visceral.
Oh, so both of them secrete this hormone?
Yeah. Here's percent fat, right? So we have our two variables, U and F. So first I'm going to talk about how food intake affects fat. So this is energy balance. The more food, the more fat. And when you do experiments on rats and stuff like that, it comes out to be a straight line, like this. More food, more fat. And there's, of course, a minimum here, which is the amount of food you need to reach zero fat, starvation. And there's also a maximum you can eat, which is your stomach maximum distension and satiety hormones from the gut, etc. So we call this the energy line, because it's basically calories. And then the other line is fat inhibits eating. And that line looks like this. Again, we know this from experiments on rodents. How do you do the experiments? You feed them, let's say, with a tube for a few weeks until they're very fat and see how much they eat the next day. Or you starve them, how much they eat the next day. All right? This is what we call the appetite line. Or people who know systems, dynamical systems, there are null lines. You clamp one variable and see the steady state of the other.
So according to this, if like I'll gain now 20 pounds, then I'll like want to eat, like yeah.
Now, where these two lines cross is the important point. This is what's called the set point, where the two lines cross, because it's the level of food that gives me an amount of fat that gives me the appetite for that level of food. It's self-consistent. If I eat more for a while, I'm going to get fatter, and then I'm going to eat less. If I eat less for a while, I'm going to get thinner, and then I'm going to eat more. So the way set point is defended, stable. And it's very amazing that we eat 1 million calories a day, a year, 1 million calories a year. And imagine how fine the balance needs to be. So over that year, we don't gain a kilo, usually. At least until you reach 50. So it's one of those amazing things. Yeah.
There is a rise in obesity in the Western world, as far as I understand from the media. How can...
Yeah, okay, so you're asking how can we analyze things like rise of obesity. So that's what I want to do. And it's true that obesity, I think, tripled from 1960 to 2000, something like that. And now the majority of people are overweight in the Western world, and like there's, 30% obesity. It's like amazing high numbers. Okay, So let's analyze. Use this to analyze, right? So I'm going to just write the equation for the energy line because it's fine. It's funny. This is change of fat. So why does fat change? It's because of the food we eat, right? Produced by the food we eat. So this is U, and there's an exchange rate of how a gram of food adds to fat. So this has to do with the composition. The more fatty it is, the more... Okay. And then there's the cost. So there's the energy cost of the muscles, etc., and basal metabolic rate, which, as I said, is unconditional love. It's the energy just to exist. Right? Sorry, that's the cost of your muscles and all this stuff. And what I meant to say is the activity plus your exercise and everything. And there's an important term here, which is the metabolic cost of fat, because fat tissue also needs to be supported. And how do we calculate the energy line? We clamp food, U, to a certain level, and ask what's the steady state fat. So we clamp food. Let's say we do a strict diet and let fat reach its steady state. So at 0 equals AFU minus B minus CF. So this line is F equals AF over CU minus B over C. This fat is a function of food, is this line. This point here is basal metabolic rate activity divided by the price of fat tissue. And the slope here has to do with the food composition. So let's see what happens if we eat fatty food. Or let's do it slow. Now, last lecture, we talked about exercise. What happens when we exercise? How does this change? Will it change the appetite line or the energy line? The energy line, because each gram of food you eat now goes also into your running activity or whatever. So how is it going to change? Cost of activity. So when you exercise, we have more of this. So that means B is higher. So this point moves here. And the energy line shifts like this. It makes sense because a given gram of food will now make less fat than before because we use it for running. Did I explain myself? This is exercise. What happens to the set point? The prediction is it moves in a funny direction down here where you lose fat, but you eat more. And that actually happens when you do look at mice and give them a wheel to run. They lose fat, like 30% fat, and they eat 20% more. Maybe you know this from your experience. In general, anything you do to the energy line is going to have paradoxical effects. Either you lose fat and eat more, or you gain fat and eat less, because it's sliding across a decreasing line. Another situation like this is hyperthyroidism. If you have a disease, you have too much thyroid hormone, your basal metabolic rate goes up, you feel very hot. This line moves here. It moves here because basal metabolic rate increases, each gram of food makes less fat, and you lose fat and eat more. So people say, doctor, I don't know why. I eat a lot, but I'm losing weight. They say, let's get a TSH test. You probably have thyroid, high thyroid hormone. All right, so that's one way to analyze how exercise can affect our physiology. Now let's talk about the appetite line. What I love about this is that you can reach conclusions so quickly. Okay. What happens if we give mice the same food but color it in many different colors. What do you think they'll do? Eat more. It's more interesting for them. It's called the buffet effect. It shifts the appetite line. So for a given amount of leptin or fat, your brain processes information and eats more. So what's going to happen to your weight set point? Eat more, gain fat. Also, processed food. Also, sugar. It should relate to the experiments I talked about here, where people with processed food or high carbs, you can say, ate more and gained weight. And that's why we can actually measure these lines, right? By experiments like that. We can measure, at least near the set point, the slopes of these lines. And by the way, in mice, it really looks like this. And it's very sharp here, and we think this is like a defense against starvation. So when your fat is very low, you lock on to the maximum amount of food. And also in here, it looks this. If your fat is very high, it's also a protection mechanism. So why did natural selection? It's clear why you don't want to lose your fat, right? Because then you starve. But why not have like a lot of fat? So imagine a big mouse can't escape, and it's very delicious. So looks like there's, in fact, in humans, there's one hormone makes you more hungry, and 7 hormones that make you satiated. One of them is called GLP-1. It's an interesting hormone. It senses food in your gut. It says, "Oh, we've eaten," and sends messages to your beta cells. Start secreting insulin. It also sends messages to your brain. Eat less. And this GLP-1 is like a miracle drug for obesity. So when I gave a course, Hormone Circuits, in 2021, I ended it with a song saying, "So what can you do if you can't diet?" Because diets didn't work, right? "And we don't have a weight loss pill." Because in 2021, I didn't know about Ozempic. Then I said something like, all you can do is walk the golden path, and something like, embrace your set point and learn to love your weight. But now, better living through chemistry, there is a drug that works on those hormones, receptors, not leptin, a different one. GLP-1 also restricts appetite, and it has a receptor, of course, on the cells, and there's a receptor agonist called Ozempic that it's like GLP-1. Basically, it makes you eat less. And people can lose, I put a graph in the lecture notes. You can lose like, I don't know, 20% of your body weight in a year. And it's now the 5th selling drug in the world. It's become like a cultural game changer. There's a lot of criticism about it because when you lose weight, fat, you don't lose fat, you also lose muscle. And so you need to eat protein and exercise when you're doing it. Otherwise, you can lose muscle mass, which can be more dangerous than having high fat. Plus a lot of stuff that we can talk about, social implications, etc. But it's important to know about this drug, Ozempic, game changer since 2021. And what does Ozempic do? Well, you're right. give him a round of applause. Unconditional love, doesn't matter what you do.
So for the same level of fat,
Yeah.
we would eat less, we would move here.
Right. Okay, Mechot Kapayim. Is that all you can do? Precisely. And Ozempic, which is actually called semaglutide or something like that. You eat less, lose fat. Okay, great. So actually, I think it even drops your maximum, if I'm not mistaken.
How did they not think about it before?
How did they not think about it before? Because it takes 20 years to develop a drug, and 90% of the drugs fail for different reasons, side effects, or they work in mice and non-humans. It costs billions of dollars to develop a drug, and since only 90% fail, it actually costs tens of billions to develop a drug that finally hits the market, et cetera, et cetera. But this is all being accelerated with the wonder of AI, and we can expect great things. All right, listen. And now I want to talk about dynamics. What happens when you stop Ozempic?
Back to the original thing.
You do. What happens to your appetite? Back to where it was? So let's analyze, all right? So
Actually, from testimony of people who use it, it happens while you're still using the drugs if you don't keep increasing the dosage.
yeah, there's adaptation, and the reason for the adaptation, one of the reasons, is losing muscle mass. Means you have less B, and then your null climbs. The energy null climb starts moving. See, but there's other reasons too. The human body, when it's starving, also probably reduces its heat generation. Heat, so also BMR. So these don't happen in mice, do happen in humans. So there's complicated year-length effects. So in diet in general, weight is a function of time in diet. You lose weight, you're really good at it, let's say, and then you rise about 50% back. And when you stop, you rise very quickly, and sometimes you overshoot. Because of these adaptations, lower BMR and lean mass. Change your energy line to which direction? The bad direction. So you gain more fat. So this is, again, I think this kind of graphic models really can help clear the mind. I hope I'm not confusing you and explaining it clearly. Okay, where was I? I was going to talk about the dynamics. The dynamics are very important, and they're very beautiful to see with this method. Let's take our beautiful weight set point. And now let's add some Ozempic. Now, okay, so this was my original. This is my current eating. I eat less and I lose fat, right? Now I stop Ozempic. Let's imagine that the appetite line snaps back to the previous line. There wasn't these fat mass adaptations. So on the next day, I look at my fat level and I read what's the food I'm supposed to eat. That's my appetite. So I shoot here. I don't go back to my former eating level. I go to a higher level. I have an overshoot. And then the next day, I gain a little bit of fat and I eat a little bit less. And over a few weeks, come back. Because there's separation of time scales. If you remember when we talked about separation of time scales, the fat sets your appetite the very next day, but when you start eating, the fat accumulates. It doesn't grow in one day. It takes a few weeks. If you think that basically, if you're only eating fat, the amount of fat you eat is more than the amount of fat you're going to store. So if you eat a kilo of fat a day, that's a lot, right? Okay. Anyway, of course, it's not only fat. Sugar gets converted to fat. Amino acids gets converted to fat. So if I draw these dynamics, if I draw them, food intake, time. So I start here on Ozempic at U1. Here I stop Ozempic. And I shoot to U2. And then I slowly adapt back. Slowly adapt back to U0, the original. This is called overshoot. So the rule is, there are two rules to remember. One is that if you move the energy line, you have eat and fat go in opposite directions, like eat more or lose fat. If you move the diet line, eating and fat move in the same direction. If I move the diet line, there's going to be an overshoot. And if I move the energy line, there's going to be no overshoot in eating. So these are like rules. So if you see something happen to a person, you can tell, is it the energy line or the diet line that's changing? And sometimes it could be both. Like if I tell you, you go fat, but you don't change your eating, that requires both lines to move. And for instance, some hormones do that, like estrogen treatment for pregnancy and stuff like that. So you can diagnose. Okay, where are we? Weight set point, leptin, 2 arms, energy line, appetite line, exercise, leptin resistance. Oh.
Why when you move the energy line, there's no overshoot?
Let's analyze it. I'm exercising, so I'm here, right? I stop exercising. What am I going to eat the next day according to the fat line? Here, I eat. I stop exercising, right? There's no overshoot, just a... Okay, so we talked about... one last thing I think I want to say is why are some people obese? I mean, in terms of this model... If you analyze leptin and fat, you get to the conclusion that people are obese because of two main effects. One is leptin resistance, which, as I said, is an adaptation where the body becomes less sensitive to each level amount of hormone leptin, probably because of this visceral fat is a signal to tell the body something's wrong here. It's actually compounding. So people have leptin. That's why injecting leptin doesn't work for obesity. And also Umax grows. Because if you eat a lot, the stomach distension grows. That pulls the whole line. So those people differ in their Umax and their leptin resistance, and therefore weight varies a lot between people. By a factor of 3 across for a given height, you might say. All right, so with age, this is basal metabolic rate, the unconditional love of the energy you make just for existing. Around 100 kilowatts, right? Like a heater. 1/10 of a 100 watts, 1/10 of a 1 kilowatt heater. So together here, 50 people, we're like a 5 kilowatt heater. So it's actually heating the room. 20% of that is your brain. 20% is your kidney. The BMR is very high in kids. Around age 20, it reaches a plateau, 50. So until age 52, I was 85 kilos. Now I'm 55, I'm 90 kilos. I'm more active. I have more muscle mass. But my BMR is dropping. So if you think about that, what's going to happen with age to eating and fat? Huh?
You eat less and have good fat.
It's like anti-exercise. Because you have less BMR, each gram of food can be translated to more fat because you burn less of it because of your basal metabolic. And so you have more fat and you eat less, according to this.
What changes the BMR?
That's a good question. What changes the BMR? Hormones, senescent cells. Who knows? Okay, now it's time for your questions.
You mentioned before how when someone gets older, then there's a certain advantage to have a higher weight because it reduces frailty. So is this like lower BMR kind of like advantageous in that sense?
So you're saying maybe this is adaptive or advantageous because older people, their optimal BMI is higher? Yeah, good question. I don't know.
How do you experiment and measure the appetite? What do you plant and do you have an equation? Do you have an equation?
We do have an equation, which is in the lecture notes. You can solve it. The way you measure it, and also in the lecture notes, is you take the organism and you put it in a diet or some kind of situation where its fat changes from the set point for a long, like for instance, tube feeding the rats with 160% of their ad libitum diet. It's terrible, but they gain fat after a few weeks. Then you take out the tube and they eat a lot less and they lose weight. And every day you can track with high resolution this line. That's why I know its shape in rats. And I think you can also do that in humans, at least near here, and like do the first order Taylor series. Yeah.
You said that like one of the explanations for obesity is leptin
Resistance.
But like, so what are we experiencing in the West right now? Like, because there are more like there are more people who are like.
Oh, you're asking why did obesity grow so much?
Yeah.
So people don't know for sure, but one thing that changed is that there's much more ultra-processed food available over the past century. People eat, I think the number is 1,000 times more ultra-processed food than in the beginning of the century.
So how will it affect like the?
It'll move the appetite line in this direction. And then when you eat a lot and become overweight, I think there are biological mechanisms that cause leptin resistance.
So like from certain BMI point, I'll just like.
Yeah, and time, and the visceral fat, and all these lipids, and the inflammation, and stuff like that. So, yeah.
So changing food composition without changing the appetite would change AF slope?
Yeah, so if you change food composition without changing the appetite, very good. Because if you change food composition, you also probably change both, but it will change the slope. So let's say for a given gram of food, you have more fat because it's more fatty, let's say. And that should make you eat less and gain weight. That's a prediction. Huh?
In keto, you only eat like fat.
Yeah, keto, you lose weight and you eat
Very fatty.
very fatty food. So in keto, you lose weight. It's true that in keto you lose weight. It's because you shift the appetite line. I think with the theory with keto, it does many things. It's primarily you cutting the carbs, so the carbs, as I said, affect the appetite. So it's like you're less hungry. So I think it's mostly shifting the diet line, but it's also shifting the energy line. So it's complicated but possible.
So shifting the appetite line is probably more significant, both there and also.
Yeah, exactly. So I didn't ever do this exercise, but this, by the way, you know that at the end of this course, there's the final exercise, right? That I'd like you to do in pairs, or alone, but they're far more fun in pairs. And you need to choose a problem that's inside the realm of this course. So, for instance, analyzing ketogenic diet with this framework. Why somebody asked, why do trees risk of death drops with age? Like, choose something and write something using the concepts of this course. And it's up to you to be creative and enjoyable and choose something that you think is interesting. That's the final exercise. So you can start thinking about that. And you should have a partner. That's why I keep mixing you guys up or asking you to pair and share. And yeah, more questions.
I think there is, like, isn't there also a positive feedback loop that when you eat more, your stomach grows and then you feel less full and then you want to eat more?
Yes, that's right. When you eat more for a long time, your Umax grows. And that pushes the...
But doesn't it also increase your appetite?
Yeah, that's right. When your Umax grows, it pushes the appetite line to this direction. When you eat more, your Umax grows, and then the whole appetite line moves.
Which parameter is it?
So for that, you need the equation for the appetite line, and that's in the lecture notes. You'll see it in the lecture notes if you're interested. Yeah.
Two parts of one question. How, let's say, predictable is the set point, given, I don't know, I can take mice from the same genetics, same food, would they be viable or not? And probably the second question is, can we tell if I want to lose XY weight, how much exercise I need, like how, formalize this movement, or is it quantitatively or qualitatively?
Yeah.
Movement?
So the question is, first of all, how variable is the weight set point for genetically identical mice in the same conditions?
Or humans, or, can you predict? Can you look at a person and without doing, giving more food or less food, tell what is the set point protein?
I don't know the answer even about mice, how variable it is, but it's certainly data available. You can use this as a qualitative theory, like we did now, but also quantitative. So you can predict, for example, once you measure this for mice, now you give them a wheel, and I tell you they eat 20% more, and I can say their weight is going to be 30% less, and it's on the button. So for mice, it's on the button. For humans, it's just never been measured as far as I know, because this is a very new concept. So this, if you want to read about it, is in the paper by Aaron Ba in 2023 that we'll link to the lecture notes. It's called Rules for Fat Interventions or something like that. I think that looking at a person's history, you can probably get these lines around their set point. And it'll be very interesting to see how predictive it is. But then you also ask, can you look at a person, I guess you mean like their genes or something, and predict their set point. Also very interesting, because you can now like think about SNPs or DNA variants that move the set point, and maybe get some insight, maybe figure out how this feedback works mechanistically. I think once you have a framework like that, you can start testing it and go all the way. By the way, we're missing one big piece of the puzzle, which is what regulates the lean mass, the muscle mass. This is a fat mass. The lean mass is regulated both by exercise. So if you exercise, you increase your lean mass. But you also need protein for that. Because if you don't have protein and you exercise, you don't increase your lean mass. If you have protein and you don't exercise, you don't increase your lean mass. You need both of them. It's an AND gate, and that's encoded by different proteins like growth hormone, IGF-1, together with insulin, and mTOR. Plus the muscles secrete hormones like irisin that tell the body how much muscle we have. Mitochondria secrete cytokines. So it's a very interesting feedback loop that if we could figure it out, maybe we could also see how to optimally reduce weight while preserving muscle mass. Because all this muscle mass stuff is very trial and error right now. For carbs, you can wear a continuous glucose monitor, which measures your glucose over the day. Then you can say, okay, I ate a banana, oh, glucose goes high. I ate orange, glucose goes high. I ate a strawberry, oh, it doesn't move. Oh. But high glucose is bad because high glucose sticks to things. So it basically damages your blood vessels. That's the problem with diabetes. You lose vasculature, blindness, kidney disease, amputation. It also hurts neurons. Sugar above 7 millimolar, normal is 5 millimolar. It's very tight. So your peaks should be under control. So continuous glucose monitor now you can buy, and you can see what you eat from carbs point of view. For protein that doesn't exist, so that's I think a big opportunity to understand the lean mass feedback. Okay, let's take a nice deep sigh of relief. And I hope you enjoy your next meal and a long life.